Hello viewers, this is Dr. Hasanat. Welcome you all to my today's lecture on carbon dioxide transport. At the same time, I will try to make you understand chloride shift, reverse chloride shift mechanism, Bohr effect, Haldane effect, and their importance. So let's have a look on the screen. All of you know that arterial blood is poor in carbon dioxide concentration. So here you can see that 100 ml arterial blood contains 48 ml of carbon dioxide. So the partial pressure of carbon dioxide of arterial blood is less and it is equal to 40 millimeter of mercury. While the venous blood is rich in carbon dioxide and 100 ml venous blood will contain 52 ml of carbon dioxide. So the partial pressure of carbon dioxide of venous blood, it is equal to 45 millimeter of mercury. That means under normal resting condition, 100 ml blood while passing through the tissue capillary can remove 4 ml of carbon dioxide. Okay, and this 4 ml of carbon dioxide, it will be transported from tissue to the lung in different chemical form. From here, we can also calculate that how much carbon dioxide will be produced by the tissue in 1 minute. Just I have shown here that if the 100 ml of blood can remove 4 ml of carbon dioxide from the tissue, then how much carbon dioxide will be removed by 5000 ml or 5 liter of blood while passing through the tissue? And you know that is the cardiac output in every minute. That is the amount of blood that will pass through the tissue capillaries. So you can easily calculate and you will find that the carbon dioxide produced in every minute, that is 200 ml per minute. Okay. Now, here you can see the real picture of carbon dioxide transport and what actually happened in the tissue level. Okay. So as you know that carbon dioxide is constantly being produced within the tissue as a result of aerobic respiration. So the partial pressure of carbon dioxide within the cell, it is maximum, that is 46 millimeter of mercury. But outside the cell or in the surrounding interstitium, the partial pressure of carbon dioxide, it is 45 millimeter of mercury, that is 1 millimeter less than the intracellular partial pressure of carbon dioxide. So the carbon dioxide, it will diffuse down to its partial pressure gradient from cell to the surrounding interstitium. So that is the very first step of carbon dioxide transport. Now, as you have seen here that in the interstitium, the partial pressure of carbon dioxide is 45 millimeter of mercury. And the blood coming to the tissue capillary from the arterial side, having a partial pressure of carbon dioxide is 40 millimeter of mercury. That means it's 5 millimeter less than the partial pressure of carbon dioxide in the interstitium. So what will happen? Again, this carbon dioxide will diffuse from interstitium to the plasma down to its partial pressure gradient of 5 millimeter of mercury. Okay. So the next step, this carbon dioxide, it will enter into the red blood cell. Okay, across the red cell membrane, as it is a very much lipid soluble substance. Now, all of you know that these red cells are very much rich in carbonic anhydrase enzyme, or carbonic anhydrase enzyme are abundantly present within the red blood cell relative to plasma. Okay, in plasma, the carbonic anhydrase enzyme concentration is very less or it is almost absent. Okay, so what will happen? In presence of carbonic anhydrase enzyme within the red blood cell, this carbon dioxide molecule loves to bind with the water molecule to form carbonic acids. So as it is a weak acid, it will be rapidly dissociated into hydrogen ion, that is the acidic molecule, and this is the basic ion, that is the bicarbonate molecules. Okay, so what will happen? As there is an excess accumulation of hydrogen ion, it is the acidic ion, it may alter the intracellular pH of the red blood cell. So it will be rapidly buffered or it will be neutralized by the hemoglobin buffer. That is a very, very important intracellular buffer present within the red blood cell. Now what happened to this excess base, that is excess bicarbonate that is constantly being produced within the red blood cell? Okay, actually, basically this red cell membrane contain a special transporter protein or carrier molecule that is also known as band 3 protein called bicarbonate chloride transporter. Okay, and that will transport this excess bicarbonate from the red blood cell out of the red blood cell to the plasma. Okay, and at the same time, as here you can see that hydrogen ion, it is a positive ion and it has been retained within the red blood cell by binding with hemoglobin. But this negative charge molecule, that is bicarbonate, it is continuously getting out. So what will happen? There is excess positive ion build up within the cell in comparison to negative ion. So for the maintenance of the electrical neutrality or ionic balance, this carrier molecule also transport chloride ion from plasma to the red cell or into the cell, and thereby helps to maintain the ionic balance within the red blood cell. Okay, so this counter exchange of bicarbonate with the chloride molecule across the red cell membrane that happens in the tissue capillary, this transport mechanism is known as chloride shift mechanism, where you can see that bicarbonate ion is getting out of the cell and at the same time chloride ion is getting into the cells. So what is the importance of this chloride shift mechanism? Here you can see by removing this excess base from the RBC, it helps to maintain the intracellular pH, and at the same time that supply the bicarbonate ion into the plasma. And you know the bicarbonate is a very important blood buffer. Okay, and this chloride shift mechanism supply this bicarbonate to the plasma. It is one of the most important source of bicarbonate. And another important source, you know, that is the kidney. From the kidney, this bicarbonate ion, it will be reabsorbed. Okay, so this chloride shift mechanism, they are efficient or they are helpful to maintain both intracellular pH and the extracellular pH, that is the pH of the blood. And at the same time, it helps to maintain the ionic balance, just I have told you, within the red blood cell. Okay. Okay. Another important point I want to mention, that here you can see this bicarbonate form. This is a very important chemical form of carbon dioxide. That is how carbon dioxide is transported in the blood in the form of this bicarbonate. So this chloride shift mechanism also helps in the transport of bicarbonate. So these are the main important points. Now, so this is the major pathway by which carbon dioxide is converted into the bicarbonate molecule, and majority of the carbon dioxide, it will be transported in the plasma in the form of bicarbonate. Okay. Now, some of the carbon dioxide molecule that will directly react with the hemoglobin molecule within the red blood cell to form carbamino hemoglobin. And basically this carbon dioxide will bind with the amino group of the hemoglobin molecule. Okay, to form this carbamino hemoglobin. Okay, so now the some of the carbon dioxide molecule also binds with the plasma proteins to form carbamino protein. Okay, so these two form, that is carbamino hemoglobin that is within the RBC and carbamino protein in the plasma. These two form together. Together they will be considered as carbamino compound, and 23 to 30% carbon dioxide will be transported in this form. But among these two forms, this carbamino hemoglobin, this form is important because the hemoglobin concentration is greater than the concentration of plasma protein. And the remaining carbon dioxide, that is very minimum, 5 to 7% of carbon dioxide will be transported in the plasma in dissolved state, or they will remain dissolved in the plasma. Okay, so these are the fates of carbon dioxide after entering into the blood. Okay, and all these forms of carbon dioxide, they will be transported from tissue to the lung. Another point I want to mention here, that here you can see as more and more carbon dioxide entering into the red blood cell and produce hydrogen ion, and this hydrogen ion is ultimately binds with the hemoglobin molecule or buffered by the hemoglobin molecule, makes the hemoglobin molecule acidic, and acidic hemoglobin has a less affinity for oxygen. So at the tissue level, what happens? At the tissue level, as carbon dioxide enter into the red blood cell and finally produce this hydrogen ion, and this hydrogen ion, it will be tied up with the hemoglobin molecule, makes the hemoglobin molecule acidic, so reduce its affinity for oxygen. So that will enhance the delivery of oxygen to the tissue, or enhance the delivery of oxygen to the tissue. And this effect is known as Bohr effect, that happens in the tissue level or in the tissue capillary. Okay, that is the, and as you know, there is oxygen delivery to the tissue increases, that will shift the oxyhemoglobin dissociation curve to the right. So what is the importance of Bohr effect? Bohr effect is important for the transport of oxygen or enhance oxygen delivery to the tissues. Okay. Now, what is the definition of chloride shift? Just I have explained. This chloride shift or hamburger phenomenon is the exchange of a chloride ion for a bicarbonate ion across the red cell membrane. Again, you can see the figure there. This bicarbonate ion, it will be counter exchanged with the chloride molecule across the red cell membrane, where the bicarbonate ion is getting out of the cell and this chloride molecule is getting into the cell. Okay. So what are the importances of this chloride shift mechanism? I have already mentioned. Just you can see here. So it helps to transport carbon dioxide in the form of bicarbonate. I have explained in the previous slide, and helps to maintain the pH of the blood. Not only blood, it also it helps to maintain the pH of both intracellular and extracellular pH. Okay, and also helps to maintain the ionic equilibrium within the red blood cell. I have explained it already. Is it okay? And this chloride shift occurs at the level of tissue capillary or at the tissue level. So the very first step of carbon dioxide transport, that is the diffusion of carbon dioxide from cells to the interstitium due to 1 millimeter of mercury partial pressure difference. Okay, I have already explained this. And then from the interstitium, this carbon dioxide, it will be uptaken by the blood or taken up by the blood in the tissue capillaries due to 5 millimeter of mercury pressure difference, because you know the blood coming to the tissue capillary from the arterial side having a partial pressure of carbon dioxide is less 40 millimeter of mercury, and you know the interstitium, it is 45. So there is a difference of 5 millimeter of mercury pressure gradient of carbon dioxide. So that will allow the diffusion of carbon dioxide from interstitium to the plasma. And now after entering the blood or plasma, this carbon dioxide, it will be transformed into different chemical form. Just the thing I have already tried hard to make you understand. So I have already shown you that 5 to 7% carbon dioxide that will remain in the dissolved state, or that will be transported in the dissolved state, and that is equivalent to 0.2 ml, because you know 100 ml carbon dioxide remove 4 ml 100 ml blood remove 4 ml of carbon dioxide from the tissue, and among these 4 ml, 5 to 7% of carbon dioxide that will be transported in the dissolved state, and that is equivalent to 0.2 ml. Okay, and the most important or majority of the bicarbonate carbon dioxide molecule, or most of the carbon dioxide molecule, it will be converted into the bicarbonate form. Okay, and that is it is actually 63 to 70% carbon dioxide will be transported in this bicarbonate form, and that is equivalent to 2.5 to 2.8 ml among the 4 ml of carbon dioxide. Okay, and you have already known that some carbon dioxide will also be transported in the carbamino compound. Okay, they are carbamino hemoglobin and carbamino protein, 23 to 30% or equivalent to 1.2 ml. But among these two forms, carbamino hemoglobin, this form is most important because you know that hemoglobin concentration is higher than the plasma protein concentration. Is it clear? So these are the three basic chemical forms of carbon dioxide by which carbon dioxide molecule it will be transported from tissue to the lung. Is it okay? Now this whole blood and these different forms of carbon dioxide it will be transported to the pulmonary capillary, or it will reach the lung level. Now let's see what happened in the pulmonary capillaries. Here you can see this is the alveoli having a partial pressure of oxygen 100 millimeter of mercury, and the carbon dioxide partial pressure is 40 millimeter of mercury. Now the mixed blood coming to the alveolar capillary or through the coming to the or reach the pulmonary capillary having a partial pressure of oxygen, it is very less, it is 40 millimeter of mercury. So there is a big pressure gradient of oxygen, and that will allow the diffusion of oxygen from alveolar air to the blood. And in the blood, this oxygen molecule will enter into the red blood cell. Now let's see what happens. Within the red blood cell, this oxygen molecule will bind with the hemoglobin molecule to form oxyhemoglobin. Now this hemoglobin becomes oxygenated. But you know, from the tissue capillary, this hemoglobin molecule binds with binds with the hydrogen molecule. Now this binding of oxygen with the hemoglobin molecule, it will displace this hydrogen ion from the hemoglobin molecule. Now this hydrogen ion become free within the red cell cytoplasm. So what will happen? This as oxygen is oxygen binds with the hemoglobin molecule, it will displace the hydrogen ion, makes the RBC cytoplasm acidic. Now this hydrogen ion will react with the intracellular bicarbonate ion to form these carbonic acids. And you know this carbonic acid, it will be dissociated into carbon dioxide and the water molecules. So now what happened? As this intracellular bicarbonate are used for this reaction, or okay, so what will happen? The intracellular bicarbonate concentration will fall down, and that will attract the entry or more carbon dioxide from outside to inside. So now there is again this carrier molecule present in the red cell membrane that will transport bicarbonate ion moves back to the RBC. Okay, and that is how this from the plasma, now this bicarbonate ion is transported in the reverse directions. And finally react within the RBC, it will react with the hydrogen molecule to form carbonic acid, and it will be transported, it will be revert back to its initial form, that is the carbon dioxides. Okay, so at the same time, this carrier protein is transporting bicarbonate molecule into the RBC, and at the same time it will remove the chloride ion outside the RBC. That means this chloride ion moves back to the plasma. Okay, so this is the completely reverse reaction occurs across the red cell membrane in the pulmonary capillary. And this counter exchange is known as reverse chloride shift, because you know in the tissue capillary, carbon dioxide is getting out and chloride ion is getting in, but in the pulmonary capillary, bicarbonate is getting into the cell and chloride ion is getting out of the cell. So this reaction occur in completely reverse direction. That is why this is known as reverse chloride shift. Okay. Now what will happen? You know this venous blood is rich in carbon dioxide having a partial pressure of 45 millimeter of mercury, and within the alveoli this partial pressure is less, that is 40. So that will allow the diffusion of carbon dioxide from blood to the alveoli. So that is how carbon dioxide will be eliminated. from the blood. So now I want to mention one point. Here you can see this reaction as actually triggered by the binding of oxygen with the hemoglobin. If oxygen molecule do not bind with the hemoglobin, this reaction will not occur. So carbon dioxide cannot be eliminated from the blood efficiently. So that means this effect is known as Haldane effect. That is binding of oxygen molecule with the hemoglobin tends to displace the carbon dioxide from the blood. Simply, this effect is known as Haldane effect. Okay. So if oxygen molecule do not bind with this hemoglobin molecule, there will be no reverse chloride shift. This so this bicarbonate ion cannot be converted into carbon dioxide. So that cannot be eliminated from the blood. So that is very important. So all this, the reaction, it is actually become reversed in the pulmonary capillary. In the tissue capillary, carbon dioxide is transformed into the bicarbonate form. But here you can see in the pulmonary capillaries or at the lung level, bicarbonate ion is reverted back to its initial form, that is the carbon dioxide form, and finally eliminated through the lung. So is it okay? So now here you can see what is the reverse chloride shift. So it is the process by which chloride ions are moved back into the plasma from RBC in exchange of bicarbonate ion. Okay, and it occurs in the lung level or at the level of the pulmonary capillaries, where the chloride shift occurs in the tissue capillaries. Okay, and just you have seen that this reverse chloride shift mechanism helps in the elimination of carbon dioxide from the blood. Okay, and I have already explained what is Haldane effect. This binding of oxygen with the hemoglobin molecule tends to displace carbon dioxide from the blood. This effect is called Haldane effect, and just you have seen this Haldane effect helps to promote the transport of carbon dioxide or elimination of carbon dioxide from the blood. While you have seen this in the tissue level, this Bohr effect promote the oxygen delivery or transport of oxygen to the tissues. Okay, so that is Bohr effect that helps in the transport of oxygen. But this is the Haldane effect. This helps in the elimination of carbon dioxide and carbon dioxide transport. So that is the importance of different importance of Haldane effect, Bohr effect. Okay, chloride shift that happen in the tissue capillaries, reverse chloride shift happen in the pulmonary capillaries. So the final steps of carbon dioxide transport that you have already understand, that diffusion of carbon dioxide from the pulmonary blood into the alveolus due to partial pressure difference of 5 millimeter of mercury. As you know, this venous blood contain more carbon dioxide having a partial pressure of 45, and within the alveoli the partial pressure is less because carbon dioxide is continuously being washed out through the pulmonary ventilation and replaced by the incoming fresh air. That is why the partial pressure is less within the alveoli. So that will allow the diffusion of carbon dioxide from blood to the alveoli, and thereby it will be eliminated from our body. So thank you. That's all for today. If you like my video lecture of or if you find this video useful to build up your academic knowledge, please subscribe my channel, share this video with your friends. So stay connected. Hope I will be back very soon with a new video. Thank you very much.